everyone. Thank you very much for joining us in the second talk from the Physio webinar. This is an event organized by graduate students from the Physiology graduate program of the Institute of Bioscience of the University of São Paulo. This is our team. We organized 4 very interesting talks with speakers from different countries talking about different physiology themes and their intersection with other areas. Please follow us to get more information so you do not miss any of the talks. Today, we are very pleased to welcome Dr. Gareth Leng, a neuroscientist who graduated in mathematics from the University of Warwick, master in neurocommunication and doctorate in physiology at the University of Birmingham in the United Kingdom. Currently, he is a professor in experimental physiology at the University of Edinburgh, a fellow of the Royal Society of Edinburgh, and an honorary member of the British Neuroendocrine Society. He published more than 300 research papers and reviews in the area of neuroendocrinology. In addition, he has published some books, one of which is the title The Heart of the Brain, the Hypothalamus and its Hormones. Dr. Gareth Leng, thank you very much for joining us today. After your talk, we will have 30 minutes to take questions from the audience. Everyone can ask questions on the YouTube chat. It can also be in Portuguese if you want. Our team will select them and send them to me. This event will be recorded and will be available on YouTube after, and with no more delays, I give you the word. Thank you.
Thank you so much. It's a great pleasure to be here. I just wish I could be with you in Brazil, but here I am in Edinburgh, supping my glass of Scotch whiskey. And it feels strange in this world that here I am just talking to myself and talking to a face on the screen. But maybe that's the nature of the times. Anyway, it's a great honor to be invited to talk to you, and thank you very much for that. I should have to say that I should thank you now, because you may not want to thank me at the end.
No,
it's always best to get that out of the way first. Okay, so yes, I have written a couple of books, and this is The Heart of the Brain, which I wrote. It was published in 2018, I think, and and that was really my homage to neuroendocrinology. I mean, I've been a neuroendocrinologist for 40 years, something like that. Yeah, and I've now come to about the end of that time, still writing a bit, but it's been an exciting journey for me, and so that was that book. I have written another one, which I'll introduce briefly, that was what came out last year, called The Matter of Facts, which is more of a kind of acid reflection on the nature of science today. It's a different style of book. Anyway, shall I just share my screen and go into this talk? So if you give me a second while I do that. And is that okay for you? Right. So yes. Here I am. That's great. Thanks. So again, this is really just to remind me and to point out to you that I certainly don't do anything on my own. And this is my current team at Edinburgh, or at least my very recent team. And I think one of the things that about any team in science is worth pointing out is just how international they are. Shireen Hassan is from Egypt, and she's just gone back to Egypt. Catherine Hume is doing her postdoc in Canada. Mike Ludwig, my longtime collaborator, he is German, he's here in Edinburgh. Tristan and Rodri are my 2 sons. And in fact, if the book on the left was written with one of them, Rodri, who is a social scientist. Jorge Marcos Roy, he's Spanish. Luis Paiva is Chilean. Nancy Sabatier is French. It's the nature of science that it has no frontiers, no boundaries. So anyway, what am I going to talk about? Let me see if I can move this down. Having said that, I have lost my screen here. Just a minute. That's interesting. Right, if I go. Okay. I have a slight technical problem here, which is I'm going to have to.
Excuse me, Gareth.
Yeah.
You are in the last slide of the presentation. You can. That's the problem. So
good. That actually makes a.
Okay.
All right. Thank you very much. that was just a deliberate mistake. I put the same slide in at the beginning and at the end. That completely confused me. Right. So thanks. I'm going to start by talking about this book. It's another book. I didn't write this. I had nothing to do with it. This was written by Jeffrey Harris in 1955. I was just 2 years old at the time, so I had absolutely no influence on it, to my knowledge. This book was a monograph by Jeffrey Harris. Jeffrey Harris is sometimes called the father of neuroendocrinology, really because of this book. It was, it's still a wonderful book to read. But he really founded the whole field of neuroendocrinology with this monograph. What was so startling about it? Well, it was something very, very simple, really. And this is one of the illustrations from his book. The book, in some ways, turned our understanding of the pituitary gland completely around. Until his monograph, it was kind of understood that the anterior pituitary was autonomous. It wasn't really controlled by the brain, or only very slightly, or only by a few neurons that innervated it. And primarily, the pituitary worked by interactions with peripheral endocrine organs. In other words, endocrinology was seen as a topic by itself, nothing really to do with neuroscience. It was merely an accident that it happened to be hanging off the base of the brain. Well, Jeffrey Harris, of course, really proposed that the anterior pituitary was controlled by the brain from the hypothalamus by releasing factors that were released into the pituitary portal vessels. Now, this, I'll come on to the implications of this and why it was so controversial, but this was a major change, because what he did was to say that, well, actually, if the brain is in control, if the pituitary is under the control of the brain, then to understand the endocrine systems and to understand reproduction, we actually need to turn to neuroscience. We need to go to a new community, use completely different techniques and completely different approaches. And that was not a very comfortable message. And this character in particular took enormous offense at it. Now, this character on the left here is Sir Solly Zuckerman, who was by far the most powerful and influential scientist in Britain at the time. In fact, not just in Britain, his influence was really global. During the war, he had been the chief scientific advisor, not only to the British government, but also to the American government. He was the chief scientific advisor to the Allied command. And after the war, he was in Britain a hugely influential member of the establishment, in control of the Medical Research Council, and also had a very visible public persona. He was very, very well known and was in every important government body. But by training, he'd been a reproductive endocrinologist. And so he really took a battle with Jeffrey Harris. And he took battle in a review that he wrote. He reviewed the book for Nature. And the review was strikingly mean, really unpleasant. If you think that scientists today can be aggressive, you haven't seen anything until you've read the review by Sir Solly Zuckerman. It started with this contemptuous sentence: the place taken by the hypothalamus in current biological discussion is in striking contrast to its size. Now, the contempt is oozing from it from the very beginning. And of course, he was pointing to something that's absolutely true. Here below that in red is about the size of the human hypothalamus, pretty tiny compared with everything else. And he went on to rail about that those who are interested in the hypothalamus seem to imagine it can do so many functions. More, it's impossible to conceive that a tiny little scrap of tissue can be involved in so many discussions. And as for Jeffrey Harris's idea that there would be releasing factors, this was really greeted with contempt. The speculations about the number of chemotransmitters that might be involved when not even a single one has yet been identified. And more than that, he said that he didn't even conceive how it could be possible to identify these chemotransmitters in such tiny amounts. Well, the arguments and debates between Zuckerman and Harris are kind of now a matter of kind of legend. There's so many stories about them. What is certainly true is that Geoffrey Harris took the review, framed it, and it occupied pride of place in his own laboratory in London. But of course, Geoffrey Harris won the argument. Time proved him right. There are all the releasing factors that he'd predicted. There's GnRH, somatostatin, TRH, CRH, dopamine, which controls prolactin secretion, and GHRH. They were all, over the next 20 years, discovered, sequenced. Everyone that he'd predicted was discovered. And Shalley and Gilliman, of course, won Nobel Prizes for discovering the first few. But the revolution didn't stop there, because it became clear over the years that followed, was that these releasing factors most of them are peptides, and the hypothalamus is actually filled with a huge number of different populations of peptide-containing cells, each peptide in a different population, some populations of cells containing many different peptides. So the hypothalamus, as we understand it now, is this myriad of diverse neurons, diverse chemical neurons. Now that was a real shock to the system. And also, if you remember what I said, that Solly Zuckerman said how impossible it was to imagine that a tiny part of the brain could be involved in so many different things. Well, I'm just going to take you through one tiny part of a tiny part. This is the arcuate nucleus in the hypothalamus. And these neurons here in green make growth hormone releasing hormone. They control growth. And there they are in the arcuate nucleus. And in the same part of the brain, these are the dopamine neurons that control prolactin secretion. And in the same nucleus, these are the kisspeptin neurons that ultimately control all of reproductive functions, at least the pulsatile secretion of luteinizing hormone. And essentially, these neurons, being sexually dimorphic, are responsible for the sexual dimorphism of males and females. In the same part, these neurons are neuropeptide Y neurons, and they make another peptide as well, AGRP. And these neurons are key to control feeding. They're not neuroendocrine neurons, but they're fundamentally important for a major physiological behavior. And here, there they are. They're not only peptide-containing neurons, but they're also sensitive to peptides secreted from the periphery. And this is the distribution of leptin receptors, all in the same part of the brain. And here we've got alpha MSH. Now, if neuropeptide Y is controlling feeding, alpha MSH almost does the converse, because that controls satiety. But it's also involved in controlling sexual behavior. So what you see is packed into this tiny part. There are many different populations that seem to do very, very different things. And some of them seem to do more than one thing. Alpha MSH is a great example of one that seems to have a key role in controlling satiety and sexual behavior. And this poses a huge challenge to our understanding. If we run through the hypothalamus as a whole, of course, we now know that the hypothalamus isn't just involved in vegetative functions, it's involved in what we might call affiliative behavior. But if you look at this photograph, it's a very famous photograph from the liberation of Paris. And if you look for a moment at this photograph, if you look at it through the eyes of a physiologist and think of what you see in that photograph, you'd forget about the boy kissing the girl. Look at the position of the girl, the head thrown back, the curved back, the raised buttocks. This is the classic lordosis posture. It's a posture that is common to many mammals, and when you see it in a female mammal, it's an indication of sexual receptivity. Now, I mentioned that because that lordosis reflex is controlled by one part of the hypothalamus, the ventromedial nucleus, and it's controlled by the actions there of oxytocin. And I'm going to talk a lot more about oxytocin in what follows. But also the hypothalamus is involved, as I said, in sexual differentiation. Again, I mean, this image of the kiss it's not really an erotic image. It really contrasts the different contours of the male body and the female body, and the shaping of the body. That sexual dimorphism is of course under the control of the hormones secreted from the hypothalamus, and that in turn control the pituitary secretions, that in turn control the gonads at the head of it, of the neuroendocrine systems. Of course, another part of the hypothalamus controls all of our bodily rhythms. It's the master circadian clock in the suprachiasmatic nucleus. And of course, I've already mentioned growth and how that's controlled, but also feeding behavior, and not just feeding behavior, but the neurons that are involved in feeding behavior are intimately tied up with the control of reproduction as well, and at a whole range of different ways and a whole range of different levels. And of course, stress, the stress axis is there in the hypothalamus. Now, and all aspects of reproduction, from sexual behavior, penile erection, coitus, sexual receptivity, all the changes that occur in parturition and in lactation, the delivery of the young, and the maternal behavior that follows, they're all governed by the hypothalamus. And actually not just by the hypothalamus, just one system is involved partly in everything that I've mentioned, and that system is the oxytocin system that I'm going to talk about in the rest of the talk. Of course, it doesn't do it on its own. But again, here is the challenge. You have a single system in a part of the hypothalamus, a part of the brain, which is incredibly complex, involved in so many different things, and this one system seems to be involved in almost all of them. And we're not talking about, and we are talking about something that is conserved through evolution to a phenomenal degree. Essentially, the hypothalamus is the same in all mammals, and indeed, it's pretty much the same in all vertebrates. There's a phenomenal conservation, and you can trace the evolution of these systems way back to the beginning of the origin of bilateral mammals. These are very, very ancient systems. They're not the products of a late spurt of evolution. They go deep into our evolutionary history. So if Geoffrey Harris has thrown the challenge to the neuroscientists to adopt this challenge of not only taking over endocrinology, but understanding really everything that's important about physiology. Now, when I say everything important about physiology, obviously I'm speaking as a neuroendocrinologist. Not everybody would agree with me, but let's just reflect for a minute on the things that are really important to us and for our health. And if the stress system isn't important, if our body composition and appetites aren't important, of course they are. They're not only key to the importance of our health and well-being, they're also our appetites and our desires, our passions, our instincts. Actually, these are things that make us human. And so the hypothalamus is really at the heart, I believe, of what really is important and makes us human. So it's a shame that neuroscientists ignored us. I think they've missed a trick there. But why did they ignore it? Why weren't they interested? Well, they weren't interested in for two reasons. Firstly, the hypothalamus was just too complicated. It's a small part of the brain with so many different systems in it. And neuroscientists really like working with big areas that were monotonously similar. But they also weren't interested because they didn't get peptides at all. To the classical neuroscientists, peptides were a peculiar class of neurotransmitters, and probably not a very important one. For them, information processing was just neurotransmitter release at synapses that's coupled to electrical activity. Everything was about that. They believed that the electrical activity of neurons encoded very specific information. Each neuron had a particular job, and the electrical activity encoded particular information to execute that task. Now, if you believe that, then you believe that the substrate of the processing is the connectome. And there are neuroscientists today that believe that actually understanding that mapping diagram of the brain is key to understanding the brain. That's the map of physical connectivity of neurons. So if you know that, we know everything there is to know. And they believe, many of them, that the plasticity of brain function, everything important about how we learn, how our brains change, consists of mechanisms that occur at the level of the synapse, activity-dependent changes in synaptic strength. And of course, we know what a number of those factors. Now, by this logic, by the logic of the classical view of neuroscientists, the brain has just got enormous processing power just because of its size and complexity. And the calculation is a simple one. Human brain's got 10 billion neurons. Each of them make about 10,000 connections with other neurons. And each of those neurons can, in principle, fire action potentials at up to 200 spikes a second. So the potential passage of information from this is 2 times 10 to the 16 calculations per second. Obviously, something of this processing power has got the capacity, they thought, to explain everything. I don't think for a minute they're right. In fact, this processing power is about equivalent to the current most powerful supercomputers that are available to us. And with the speed of advance in computation, computer science, and computer power, our desktop computers will have comparable power in just a few years' time. We're not far off that now, and I don't think for a minute that we are close to having machines that approximate the power of our brains. So we've got to look elsewhere. We've got to look in a different way to understand what our brains do and how they work. Well, coming back to peptides, let me say very clearly that peptides are not neurotransmitters. In my view, it is commonly said that peptides are neurotransmitters. I think to a first approximation, it's a lie. It's complete nonsense. Firstly, peptides are not packaged. Although peptides and neurotransmitters occur in the same neurons, they're not in the same places, and they're not in the same organelles. Neuropeptides are packaged in quite large vesicles, much, much bigger than synaptic vesicles, much bigger than those that contain neurotransmitters. And a peptide molecule is much more potent than a neurotransmitter molecule. It's about 1,000 times more potent. Neuropeptides work at G protein coupled receptors, and they have nanomolar affinity for their receptor. Conventional neurotransmitters like glutamate, they have micromolar affinity for their receptors. Now that difference is very important. If you think of a single synapse, typically when an action potential invades a presynaptic ending, you'll typically get about 1 vesicle of glutamate released, roughly 1 vesicle released at each synapse for every action potential. And that vesicle will contain about 5,000 molecules of glutamate. And now that's enough to raise the concentrations to a very high level in the synaptic cleft. And so glutamate will act at the postsynaptic membrane and nowhere else. So it's a mechanism. The glutamate receptors are not particularly sensitive, and that's important because it's important that they can only see the message that's released from the presynaptic ending. Neuropeptides are really very, very different. Not only have they got much greater affinity for their receptors, they're much longer lasting. Neurotransmitter disappear with a half-life of a few milliseconds. Peptides have got half-lives of minutes in the brain. And as we'll see, they're not really released at synapses at all. And they're hugely diverse. There are only a handful of conventional neurotransmitters, but we know now of at least 300 neuropeptides that are signals in the brain. They're different in lots of ways. And just how different they are really was a consequence of studies over 20, 30 years, more than that, of the magnocellular system mainly, this lovely model system that I'll just show you just how beautiful it is at the moment. These are the neurons that project to the posterior pituitary. And these are neurons that make these oxytocin that I've mentioned, and half of them make vasopressin. But it's the oxytocin system that I'm really going to talk about. If we take a coronal section through the rat brain, and we actually look at these systems, here they are. It's the oxytocin cells here are in red, and the vasopressin cells are in green. Now, this is the paraventricular nucleus at the top, and at the bottom, you have the supraoptic nucleus. Now, it looks pretty, doesn't it? Every one of these neurons in the supraoptic nucleus, and nearly all of them in the paraventricular nucleus, project to the posterior pituitary. Many of them project nowhere else, though some have got collateral branches, as we now know, that go to other parts of the brain. Now, The supraoptic nucleus is particularly lovely because it only has two types of neurons. Every neuron in this nucleus is either an oxytocin cell or a vasopressin cell, and every one of them projects to the posterior pituitary. So it's a very homogeneous, very compact part of the brain. And as you'll see, it's on the base of the brain, and it's next to the optic chiasm. And for an experimentalist, just think how wonderful that is, because it's a relatively simple nucleus. It's away from lots of other areas, and so you can go and manipulate it. You can take it out, you can study it in vitro, you can identify single neurons in vivo. They're big neurons, so they make tons of their product. So they're easy to see, easy to visualize, easy to manipulate. It's a fantastic model system. And because of that, we probably know more about these neurons than about any neurons in the brain. So if we have a look at those now in a little bit more detail, and just to give you a glimpse of just how and in what detail we know about these, we know now that in the rat brain, there are about 5,000 oxytocin neurons. And virtually all of them, in the rat except about 60, project to the posterior pituitary. Nearly all of them do that. Now, in the posterior pituitary, each one of those axons gives rise to about 2,000 swellings and nerve endings where the vesicles are stored and from where they can be released. And when they are released here, then they will enter the bloodstream. Now, if we have a look at the posterior pituitary under the electron microscope, we can see these. And here, highlighted here, is one of these swellings. And these dark little spots are the neurosecretory vesicles. Now, from some amazing work done in the 1970s by Jean Nordman in France and John Morris in Oxford, they did some very, very careful, detailed, quantitative electron microscopy to really describe this tissue in enormous detail. And so we know from their independent work, there's two labs going on at much the same time, that oxytocin is contained in about 7 billion vesicles in the pituitary. Each one of those vesicles contains about 85,000 molecules of oxytocin, but not oxytocin on its own. Oxytocin is synthesized as part of a large precursor. Oxytocin is only a peptide of about molecular weight 1,000. It's synthesized as part of a 23,000 molecular weight precursor. And that whole precursor is packed, 85,000 molecules of it in each of these vesicles. Now, that is a huge amount of peptide packed in a very small space. In fact, that's why these are dense cord vesicles, because they're packed in essentially a crystal-like form. It's a crystalloid form there. There's really almost no space for anything else. It's virtually a solid there. Now, if we also know, as I've indicated, in enormous detail, a huge amount about the electrophysiology of these cells and of all their electrical properties. And I'm only just going to kind of touch on a very few examples here. And this is the electrical activity in spikes per second of a single oxytocin neuron. We'll come to some of the classic stimuli later, but what you'll see here is that this is a response to an intravenous injection of cholecystokinin. And this had been known for a long time. It was really one of the earliest indications that oxytocin secretion was controlled not just by reproductive stimuli, but also by appetite-related stimuli and gut-derived stimuli. And this is very consistent. This is the average response of 36 cells down here to CCK. So it's a very typical response here. And what you see here in the same experiment is the response of the same cell to an intravenous infusion of 2 molar sodium chloride at a very, very low, slow rate. It slowly increases osmotic pressure by about 15 milliosmoles over half an hour. And what you'll see is that the firing rate of these cells tracks closely that increase in osmotic pressure. And again, here that's the group results. So again, this response has been known even longer than the CCK response. It's known that in rats that oxytocin is involved in regulating the kidney. It's a natriuretic hormone in rats. And in fact, there are some very kind of famous and well-known Brazilian scientists who've made a huge contribution to understanding the role of oxytocin in electrolyte homeostasis. I'm pointing out this here because here we've got cells already that are involved in two apparently very, very different functions. One in regulation of appetite, energy balance, and one in electrolyte homeostasis. and neither of these seem at all remotely connected with the classical roles of oxytocin, which is in lactation and in parturition. But one of the reasons I showed this is that we know so much about the behavior of these cells. We know how they respond in terms of electrical activity to a whole wide range of stimuli. And of course, in response to all these stimuli, we can also measure how much oxytocin is present in the blood, how much oxytocin is released as a result of that. So we can kind of put all of these things together. We have this detailed understanding of the pituitary. We know how many cells there are. We know how many axons there are. We know all the release sites. We know how much every hormone every vesicle contains. And we know the plasma concentration, and we know what the electrical activity is that produces that plasma concentration. So we can now run through some very, very simple calculations. And the answers, though, to those simple calculations, the answers are inescapable. It's all published. I'm not going to go through the calculations now, but it's inescapable that actually, although there are 7 billion vesicles in the posterior pituitary, then normally, under basal conditions, each cell releases just one every 5 seconds or so. Even though it's got 2,000 release sites in the pituitary, the release of vesicles is a very, very rare, very sparse event. And even under the extreme activation of dehydration, that increases the secretion more than tenfold, you still only get about 2 vesicles from each cell per second, even though there are 2,000 release sites. So we can do this calculation. At any one of those release sites, it takes on average at least 400 spikes to release one vesicle. Now that's in enormous contrast to the release of glutamate at a synapse, where one spike releases about 1 vesicle of glutamate. So in fact, this is probably a very generous calculation. Probably the true figure is closer to 1,000 to 2,000 spikes is needed to release a single vesicle from any one site. So that seems very, very sparse. You might ask why there's so many vesicles if they're only released so rarely. The short answer is there are an awful lot of seconds in a day. One of the big differences between peptides and neurotransmitters is that when a synaptic vesicle containing glutamate is released, then it can be quickly refilled from the terminals. There's reuptake mechanism, refilling mechanism. When a peptide containing vesicle is secreted, you need to make a new one up in the cell body in the hypothalamus and transport it all the way down the axon, and that takes a lot of energy and that takes a lot of time. That's why you need to maintain a large store of peptides. Anyway, that's what happens in the pituitary. And you saw how many vesicles are present in those nerve endings there. They're packed with vesicles. We can run through a few calculations. Well, we know these peptides are also released in the brain. Well, we can do a kind of thought experiment here. We know there are 5,000 oxytocin cells in the hypothalamus. And again, we know how many vesicles are present in the hypothalamus. And we know that there are 11,000 vesicles in the dendrites of these. Why we're talking about dendrites, I'll come to. Now, if just one of those is released from each cell, on average every 15 minutes, just one every 15 minutes, then we can calculate how much hormone will be released into the hypothalamus. We don't really know what the half-life of oxytocin is in the brain. We know what it is in the blood, and we know what it is in the CSF. In the CSF, it's about 20 minutes. In the brain tissue itself, it may be shorter, but it can't possibly be shorter than 30 seconds. That would be shorter than anything, any peptide half-life we know for any peptide under any circumstances. We don't think it's anything like that short, but let's suppose it was just 30 seconds. And let's suppose that what's released from these magnocellular cells is distributed in the entire volume of the extracellular space of the hypothalamus. Well, the volume of extracellular space in the hypothalamus is just about a microliter, and that's the whole hypothalamus. So what you'd get from these things, from that half-life, that volume, that release rate, is an average concentration of more than 500 picograms per ml throughout the hypothalamus. Now, that may or may not mean anything to you, but if I tell you that in the periphery, You only need about 10 picograms per mil to have the potent effects that you see in the periphery. So what does that tell us? Well, that tells us firstly that you don't need very much release of very many vesicles in the brain to have a large concentration over a pretty wide area. And what that means is that you don't need anatomical conjunction of neurons to pass messages. But if you look in the textbooks, then you will see the dogma, the classic dogma. These are the textbooks written by neuroscientists, not neuroendocrinologists. And you'll see the classic dogma of neuropeptide vesicles coexisting with transmitter vesicles at synapses. Now, we know, I've already mentioned, synaptic vesicles, you really only find synaptic vesicles at synapses. You don't find them anywhere else. But peptide vesicles are not specifically localized at synapses. You find them throughout the cytoplasm of a cell. Now, if you look at this diagram, this classic diagram, you might have seen it before yourself, similar diagrams before. What you should notice is that there's something missing from this diagram. And what's missing from the diagram is the dendrites. And actually, for the average neuron, the dendrite contains 85% of the cytoplasm of a neuron. So this diagram leaves out completely 85% of the neuron. And in the brain, most of the peptide vesicles, if they're distributed throughout the cytoplasm, most of them in the brain will be in the dendrites. You do find some vesicles at synapses, and here we are. There's some dense core vesicles, these large vesicles in this synapse at the bottom here. But how sparse they are compared to this huge number of synaptic vesicles. And please note that you don't see these close to the synaptic specialization where they've got to be released. So I don't think that release at synapses into the synaptic cleft can possibly be very common. But dendrites, though. Well, of course, to the classical neuroscientist, dendrites are the place where cells receive information. They don't transmit it. But if these are the oxytocin cells in the supraoptic nucleus, and at the base of the brain, this mat of fibers here, these are all dendrites. Now, this section has been stained for immunostained for oxytocin, and these dendrites are stained, and that is because the dendrites are packed full of oxytocin-containing vesicles. And here you can see them under the electron microscope. Here they are in a rather special configuration. Here are the dendrites lined up close to the terminal membrane, and you'll see some of them are being exocytosed. This is an image from John Morris's electron microscopy. So this is a release site for oxytocin, but is release at this site governed by electrical activity at all? Well, you might think, again, for a classical neuroscientist, what else could it be if that's what neurons do? But actually, this is results from my colleague Mike Ludwig before we started working together. This is his results in Germany, and he had reported that what he'd done was something really kind of very interesting. And what he'd done was place a dialysis probe on the supraoptic nucleus, and also a dialysis probe in the blood vessels, and that allowed him to measure oxytocin release both into the blood and from these dendrites at the same time. And he looked at that release in response to hypotonic saline. And hypotonic saline causes a rapid and strong release in the blood that follows that electrical activity, and then in a conscious animal it dies off. But in the hypothalamus, in the supraoptic nucleus, it does cause release, but it's very, very delayed and not coming out at the same time. And that's the first indication, really, that actually, yes, you can get release from the dendrites, but it doesn't follow the same rules as release into the blood. And when Mike came to work with me in Edinburgh 25 years ago now, nearly, really the first thing we set about doing was he tried to set about convincing me that the results that he came with were true. And we set about looking then at finding out exactly how oxytocin could be released from the dendrites. And I have to say that initially when he first came, I was really very skeptical. I didn't really quite believe his results. But we went about trying to reproduce them. The way we tried to reproduce them was by stimulating the axons of the cells electrically. When you do that, you invade the cells with antidromic action potentials. And if those cells are invading the cell bodies and invading the dendrites, then you should get a release of oxytocin and vasopressin if that release is activity dependent. And that's what we set about trying to do by dialysis, and frankly, it didn't work. We tried it again and again, all kinds of conditions in lactating rats. I won't go through all the things, all our catalogue of failure, but even stimulating at high frequencies for a prolonged time, we couldn't get anything out. And it was a dilemma to us. And actually, we now know why we couldn't get anything out, because normally the vesicles are not close to the cell membrane as they were in the electron micrograph I showed you earlier. They're normally right in the center of the dendrite. They're far away from the plasma membrane. That means they're far away from the voltage-gated calcium channels. However, we discovered that you can do something to change things radically. And what we discovered was that if you expose that tissue to thapsigargin to mobilize intracellular calcium stores, and you just expose it briefly and then take the thapsigargin away, you let the tissue recover for half an hour, and then stimulate again. Now you see you're not really seeing any increase following the thapsigargin in control conditions, but now if you stimulate, you get a huge amount of oxytocin being released. So mobilizing those intracellular calcium stores made those vesicles available for activity-dependent release. Now that was the kind of key breakthrough, and I'll run through here what we think happens to these cells at the moment. Normally, what you see is that in the dendrites of the oxytocin cells, the vesicles are in the center of the dendrites. So electrical activity will be propagated down the axons, will cause release from the pituitary gland, but you don't get any dendritic release. But if you have an agent that mobilizes intracellular calcium stores, then you can get some release from the dendrites. And actually, as I'm going to show you, you can get some release now without any change in electrical activity at all by peptides of the dendrites. But much more importantly, you can get some peptides can cause this margination of the granules, moving these vesicles into juxtamembrane locations next to the plasma membrane. Now, once these stores have been primed, this is a phenomenon we call priming, then electrical activity in the cells will not only cause release from the pituitary, will also cause release from the dendrites. And because the dendrites have got oxytocin receptors as well, you've now got a positive feedback system. You've got oxytocin release, you're acting on oxytocin receptors on the dendrite, you've got a positive feedback that can synchronize their activity and drive bursts. And that became very important for understanding the next thing that I'm going to talk about. Just before I do, though, that was thapsigargin, which is a pharmacological mechanism for releasing intracellular calcium. We quickly went on to find endogenous peptides that will do exactly the same thing. And this here is just example of I think the nicest example we've got, which is from a peptide called alpha MSH, which is released from the arcuate nucleus. Now, what we knew is that alpha MSH would induce FOS expression in the supraorbital nucleus. It will induce expression of the immediate early gene CFOS. Most people think when they see that, that's an indication of neuronal activity. Well, if you see FOS expression, it's really an indication that the cell is expressing FOS. Because actually, alpha MSH doesn't excite the cells, it inhibits them. If you expose the oxytocin neurons to alpha MSH, there we are, we get an inhibition of spike activity. And if you measure oxytocin in the blood, you'll see that that release is inhibited. So alpha MSH is going to induce FOS, but it will inhibit release from the pituitary. But if we look at what's released in the supraorbital nucleus, alpha MSH is inducing oxytocin release at that site. And it does that because alpha MSH mobilizes intracellular calcium stores. Here on the top, here on the left, you've got a calcium imaging signal in response to alpha MSH. This is in vitro now in the bath. you've got an increase in intracellular calcium that acts through MC4R receptors, which are expressed on oxytocin neurons. It's completely blocked by an MC4R antagonist, and it recovers after it. So alpha MSH mobilizes intracellular calcium, and that mobilization of intracellular calcium will both prime the oxytocin cells and trigger dendritic release. And at the same time, it inhibits their electrical activity and inhibits secretion in the blood. Now that is hugely important because it really shows that these two different compartments of a cell can do two quite different things. Well, why is what I've been talking about important physiologically? And here I'll go on to the classic role of oxytocin. The classic role of oxytocin is in the milk ejection reflex. And this is a painting by Tintoretto, 400 years old. It hangs in the National Gallery in London. And it, of course, shows the infant Hercules sucking out one breast of his mother Juno. And what you see is milk spurting from the breast that's not suckled. And that's simply because the milk letdown reflex is not a reflex that is local to the breast that's been suckled. It's a systemic reflex. And it's a reflex that requires stimulation of the release of oxytocin from the pituitary gland. So you have the suckling evokes an afferent pathway through spinal nerves to activate the oxytocin cells. Now, the nature of that reflex was discovered back in the 1970s by some colleagues of mine, John Wakely, who was then a PhD student. And the preparation he worked with was the lactating rat, the anesthetized lactating rat. And he set about recording from these oxytocin cells in the supraorbital nucleus and recording their activity in response to suckling. And what he observed 30 years ago really was startling and really defined the field for a generation because, well, what you've seen in the other circumstance that the oxytocin cells fire fairly quietly, just 2 or 3 spikes a second, click, 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 click. But if you put a litter of pups on to suckle at a lactating rat, then every few minutes, these cells do something dramatic. They fire in a burst like this, and this burst lasts only about 2 seconds. So cells that were going click, 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 this explosion of activity, and every oxytocin cell in the hypothalamus does burst in the same way at the same time. So you've got these intense synchronized bursts. Now I hope you'll kind of understand why I described the importance of priming in the way that I did, because that suggested a substrate, a mechanism for how these bursts occur. Now, we already knew that oxytocin was somehow involved in orchestrating this discharge through some glorious work in the 80s and 90s by Françoise Mousse and her colleagues in France. What she had shown was that if you inject oxytocin at tiny amounts intraventricularly, if you do it in a lactating rat that is being suckled, then you'll facilitate the milk ejection reflex. And this is the milk ejection reflex as seen by the intramammary pressure changes. And at the bottom here is a recording from one of her experiments of oxytocin cells and following an ICV injection of oxytocin. Now, at that time then, this was a phenomenal demonstration that really the peptides that are released within the brain are really important physiological signals. And she went on to show very quickly that it wasn't just a pharmacological effect, because if you give an antagonist instead of oxytocin, you will completely block the ongoing spontaneous reflex, even if you give an antagonist at small doses into just one supraorbital nucleus. And so with this, she'd really, I think, been the very first person to show an important physiological role for an endogenous neuropeptide. And it was such a striking role. Because what happens here with oxytocin is not something boring like you'd get with a neurotransmitter. The cells are excited or inhibited. No, you get the orchestration of a complex behavior, a complex reflex, these intermittent, intense bursts, the synchronization of behavior. It's remarkable. Well, I'm a mathematician, I think, as you heard at the beginning, and as mathematicians do, if you have a lot of data, you know a lot about the electrical properties of neurons, you can build models of them. And those models essentially translate the properties of those cells in terms of their membrane properties into differential equations. And our challenge here then, and we'd already knew that we could build very accurate models of oxytocin cells under normal circumstances, our challenge here was to find an explanation of the milk ejection reflex. And what we knew is that each of the cells, oxytocin cells, has about 2 dendrites, and that each of those dendrites are bundled together in groups. So you have a bundle containing maybe 8 dendrites from 8 different cells. And so you've got all of these different bundles shown in yellow here. And so this is the substrate of communication between the cells. They're not all coupled directly to each other, but they're all have dendrites that are coupled to some other cells. And so in our first model, you have about 50 neurons, each with 2 dendrites, and these are converging on these dendritic bundles here. So that's the topology of a network model. And then in our model, each of these cells is independently modeled by a suite of differential equations, and each has its own kind of random inputs. And what we had hoped when we were building this model was that if we simulate this kind of dendrodendritic communication that we see with priming, then what we'd see is we'd see the cells starting to be, initially they'd be completely random and independent, but then suddenly we'd hoped we'd see something like this, because this here is what a milk ejection burst looks like. This diagram at the bottom, each of these dots shows the timing of a particular spike expressed as spikes per second. So this is the intense, massive increase in firing rate you see with the burst followed by silence. This is what we wanted to see from our model. And obviously I'm only showing you this because of course that is what we saw in the model. The model every 10 minutes, 5 to 10 minutes, just synchronizes and fires one of these discharges that is, fortunately for us, almost virtually indistinguishable from the real cells. So for me, I mean, that was kind of, and for Francois Moose, we did this work together with other mathematicians. This was really the culmination of a lot of our effort over many years. So let's go back to that. In the time I have left, let's go back to that dogma that I talked about, and the idea then that the substrate of processing in the brain is this map of physical connectivity of neurons, the connectome. Well, what I've said is that peptides the peptide actions can't be bounded by physical connectivity. There's just too much abundance, and they can diffuse too far, and they last too long. They're going to spread. They don't need physical connectivity. They can't spread it infinitely, but they don't need direct apposition of neurons. And more than that, if we look at some of the other behavioral actions of oxytocin, which is not our work, I'm not really going to talk about those, but the actions on affiliative behavior. And this is the classic work from Tom Insel. Now, what he looked at was affiliative behavior, bonding behavior in prairie voles and montane voles. And the prairie vole is a monogamous species. The montane vole is a promiscuous species. In the prairie vole, the monogamous bond, is, we now know, formed by the release of oxytocin in the brain as a result of sexual activity. But you can't get that bond in a montane vole. But what's different between these species is not the oxytocin system. The oxytocin neurons are the same, released by the same stimuli, much the same way. No, it's the distribution of receptors. And this is the map of receptors in the 2 species, just showing how very, very different they are. They're in different places, in different abundance. And for the most part, they're in places in the brain where you find no oxytocin fibers at all. So the important actions of oxytocin seem to be determined by the distribution of receptors, not by the distribution of fibers. Now, I mentioned that the oxytocin system is ancient. In fact, we now know from comparative genomics that something maybe as long as 1 billion years ago, this little animal, this hypothesized animal, Ubilateria, the ancestor of all bilateral animals, had a genome actually almost as complex as ours. It had about 20,000 genes. Now, how do we know that? Well, of course, this organism doesn't exist now, but what we can say is that what comparative genomics does is to look at the genomes of vertebrates, annelids, insects, a whole range of species, and looks for commonalities between them. And if you find the same gene and the same mechanism in 2 different species, then the logic goes they must have been in a common ancestor. And it's that kind of argument that traces these back, because we know that oxytocin-like peptides are present in nearly all insects, present in most invertebrates. Of course, they're like all vertebrates have got a very similar system, but the ancestral peptides go way, way back. So from the recent work from comparative molecular genetics, It's argued that this ancestor possessed a brain-like structure that was defined by an ancestral developmental genetic program that's really quite similar to those that we see in certain neurons now. But in particular, we know that this organism seems to have expressed an oxytocin-like peptide that was present in multisensory, multifunctional neurons that are linked to reproduction and feeding behaviors to environmental stimuli and nutritional state. Extraordinary insight that the multifunctionality of these neurons is not a recent occurrence. It was really right back there a long way ago. And I've only touched on that, the multifunctionality. And our most recent work has been on those gut signals. And we now know that if you gavage with sweet food, sweet condensed milk in a rat, then you will get this lovely response of oxytocin cells. Very, very consistent. And the interesting thing about this is that this is very specific to sweet food. If you do the same thing with isocaloric cream, you get the opposite effect. You get inhibition. And so we now know that actually we can add something else to the functions of oxytocin. It's involved in glucose homeostasis in the brain. So here's the list, a partial list of things that we know oxytocin is involved in. We know it's absolutely essential for milk letdown and lactation. We know it's got an important role in parturition. That's what it's named for. We know in some species, at least, it's involved in triggering maternal behavior. In many species, it seems to be involved in social recognition, involved in sodium excretion, salt appetite, appetite for carbohydrates, energy expenditure in some species like mice, glucose homeostasis, sexual behavior, sexual receptivity, penile erection, lordosis, affiliative bonds, involved in pain processing, and the list goes on. Now, this list would be fine if you've got hundreds of 20 different populations of neurons that happen to make oxytocin and release it in different ways. You don't have that. You've got mainly, almost exclusively, one big population of cells that seem to be all very similar, do much the same things, all project to the pituitary. So you have true multifunctionality. So how does that multifunctionality arise? Well, we don't know all of the answers, but I'll just give you part of the answers, the bits that we do know. What I've told you already is that in response to suckling, oxytocin is released in these pulses, these big pulses every few minutes. I told you that in response to increased sodium in the blood, their firing rate rises continuously. So if you combine suckling and hyperosmolality, you will get pulses superimposed on a rising background. Now, you've got, so in other words, you apparently have a conflict between these signals. But do you? You don't. Why not? Well, actually, the mammary gland isn't terribly sensitive to oxytocin. Actually, the mammary gland will only see these high concentrations that you get with a pulse. So the low concentrations that you get in response to, and the small rise you get in response to hyperosmotic pressure, they're ignored. The mammary gland just doesn't see them. It'll respond non-linearly when there's a big pulse, but it won't respond to small changes. On the other hand, the kidney does the exact opposite. The kidney is very, very sensitive to oxytocin. So it does respond to the small changes that you get with hypotonic saline or with raised blood sodium, but it needs a sustained signal. Actually, the perturbations like the pulses, they don't do much for sodium excretion. You need a steady signal to activate that. So just from this signal encoding and decoding, the same cells, by the way they behave, can do two apparently different things at the same time. And then, but that's just, that's just from what's released at the pituitary terminals. And in the brain, as I've said, you can have completely independent release from the dendrites. You can have different signals organizing this, and these can have effects at diverse targets. But do they have effects at all of those targets all the time? Well, no, because actually, it's the regulation of the expression of receptors that becomes really critical here. The oxytocin receptors aren't static. They're dynamically regulated. They're there in some circumstances and not in others, and in different places and different amounts. So you have a completely different level of regulation of the functions of the neurons through regulation of receptors. So here, you'll be glad that I've come to the end now. If we go back to the central dogma of neuroscience, I described that as saying that information processing in the brain consists of neurotransmitter release at synapses that's tightly coupled to electrical activity. Well, what I've told you is that peptides don't act at synapses. They're not released from synapses. And they can be released independently of electrical activity. And in fact, when they are released in response to electrical activity, that release is very rare, very sparse, pretty stochastic. And their effects are so long-lasting that really the dynamics of release become sort of irrelevant. The classic dogma is that the substrate of processing is the connectome, the map of physical connectivity. Well, I've said that they don't depend on that peptide actions depend on where the receptors are, not on where the release sites are. The classic dogma is that plasticity of brain function consists of activity-dependent changes in synaptic strength. Well, here in neuroendocrine systems generally, peptide systems generally, we know that plasticity can occur in other ways. It can occur through priming, the mechanism that we've done, but I don't think that's the only one by any means. Probably a much more important, much more general plasticity in effects is almost certainly through effects on receptor distribution. And effects on receptor distribution in particular that are mediated themselves by the actions of peptides, because peptides don't just affect electrical activity. Probably the far more important effects of peptide signaling in the brain is effects on gene expression. And the classical dogma is that electrical activity in neurons encodes specific information. Well, clearly in the neurons that we work in, that's just as a non-starter. All the peptidergic neurons that we work on are multisensory. They respond to multiple different modalities of stimulation, and they're multifunctional. They are multitasking neurons. Now, I don't really think that neurons in the hypothalamus are that special. We now know probably that most neurons in the brain make peptides as well as transmitters. So the neurotransmitters, the language of neurotransmitters, the classical dogma is just one language. Neurons speak in many different tongues for different purposes. Of course, we don't know it all, and that the future, what we know in the future, that's down to you. I'll stop there and thank you.
Thank you very much. Now we have some questions. I will start with the fourth. Which parts of the brain segregates alpha MSH, and does it has a direct relationship with reproduction process? So
yeah. Okay, that's a very important question. Yes, alpha MSH is a product of POMC. So the precursor is pro-opium-melanocortin. So it's the same neurons make beta endorphin and alpha MSH. Now, these are neurons in the arcuate nucleus. Alpha MSH is actually made in 2 sites in the brain, but the important one is in the hypothalamus, and the bulk is made in the arcuate nucleus. And these neurons, we know, have got 2 major physiological effects. We know that alpha MSH is the most potent satiety-inducing agent that we know. It's very potent at inhibiting appetite. And we also know that it's a very, very potent stimulator of sexual behavior in both males and females. How we know that, you can ask me over a drink sometime, and I'll tell you about the story of how that arose. But there's the classical action. And one of our interests in alpha MSH were because if you look at them, the behavioral roles of oxytocin, they're exactly the same. That oxytocin is an appetite inhibitor and a stimulator of sexual behavior.
Yes, another question is about females in particular. Oxytocin has a lot of functions in females. How this functions and how these functions work in different phases, like between menstruation and
it's a very important question.
Other things, because
a very important question.
Has regulations with, as you said, regulation with kidney and other parts. How this works in a female body?
It's a very important question, and I think that a major part of the explanation is to understand that the oxytocin receptor gene has got an estrogen response element, and the expression of the oxytocin receptor is very, very strongly influenced by estrogen. We also know that progesterone and progesterone metabolites interact with the oxytocin receptor, so that both estrogen and progesterone can interact with the oxytocin receptor. And in the biggest example of this is, of course, in the uterus. In women, in the few hours before they give birth, when you're pregnant, in the few hours before you give birth, the sensitivity of the uterus to oxytocin increases more than 100-fold. And that is because of a very rapid upregulation of oxytocin receptor expression in the uterus. Now we know that in the brain, you've also, in certain populations in the brain, you've also got estrogen responsiveness of the oxytocin receptors.
And that is related with the maternal instincts and other behaviors that a mother has with infant, newborn.
That's correct. In fact, the evidence is that oxytocin can trigger maternal behavior in some species. We know it's true in sheep, it's true in rats, but in those species, it'll only work if the animals have been steroid primed first. So they have to be first primed with estrogen or estrogen and progesterone. But if they're primed with them, then oxytocin will trigger maternal behavior, but not otherwise.
The third is a little far off, but it is possible that the neurons release neuropeptides even when treated with inhibitor of electrical activity, such as CTX. So depend of.
Yes, that's right. The clearest example, which is what I tried to show, is of alpha MSH, which will actually inhibit the activity, but will stimulate release. You've got to think of peptide neurons as kind of hybrids between endocrine cells and neurons. So, of course, endocrine cells classically many endocrine cell types can release their products without an increase in electrical activity. But in some, you've got a coupling of electrical activity and secretion as well. Well, oxytocin neurons and other peptide neurons in the brain seem to have these hybrid properties in different compartments. Now, in the dendrites, you've got these calcium stores in the endoplasmic reticulum. And so the dendrites are a site at which calcium can be released from these stores and stimulate secretion. You don't have a rough endoplasmic reticulum in the nerve terminals. So in the nerve terminals, release is purely activity dependent through voltage gated calcium entry. So you've got different mechanisms, different compartments of the cell. There are other factors that will release oxytocin through mechanisms that we don't understand at the moment. It doesn't stop there. There are interesting things happening, and there are some well-established old findings in the literature that we just don't have an explanation for at the moment. I don't think we've come to the end of what we know by a long way.
I think it's this. Thank you very much. For an amazing talk. I didn't know half of this of oxytocin. I only know of reproduction part. When you talk about other things, I was like amazed. And I also want to thank the audience for their attention, the questions. I hope they enjoyed the talk. I hope you enjoyed talking with us. And we'll see you all next talk.
Thank you very much. Goodbye.